Now we're going to go into DNA sequencing and replication. So I think the most important sequencing technique that you guys really need to know for the MCAT, I think the other ones are pretty low yield, so I wouldn't necessarily focus as much on those as Sanger sequencing, but Sanger sequencing is used to determine the sequence of a strand of DNA. So to the right kind of gives a visualization of what you would be looking at. And so Sanger sequencing uses a single-stranded DNA, so that is known as the template strand. It also uses a primer, DNA polymerase, deoxynucleotide triphosphates, so these are DNTPs, and also labeled dideoxynucleotide triphosphates, DDTPs. So just also, you guys might know what a primer is already, but just to make sure, it's a small fragment of DNA that's complementary to the 5' end of the single-stranded DNA of that template strand. And so from the primer that binds first to that 5' end, DNA polymerase extends it by adding DNTPs that are complementary to the nucleotides on the template strand. And then DNA polymerase continues adding the DNTPs until it adds a DDNTP. And so DDNTPs, the reason why it stops adding is because they are pretty much the same idea as DNTPs, except they don't have that 3' hydroxide portion on the sugar. And also each is uniquely labeled with a fluorescent reporter, which is specific for Sanger sequencing. And just so DNTPs are like the A, T, G, and C nucleotides that we usually see with DNA. And so once the polymerase stops at that DDNTP, the partial strand is left unfinished, and then it kind of just breaks off like that every time this process kind of repeats. And so in order to determine like the full DNA sequence, usually there are 4 reaction containers. So for each of the nucleotides, so A, T, G, and C, and then the DD, yes, for each of the DDNTPs. And then from that, usually the distance the primers will travel is indirectly proportional to their length to determine the complementary sequence and then the template sequence. So every time, they're adding a primer and then watching where it ends, they can usually add it a little bit after from the distance that we already knew. And so as they keep going through that in each of the different reaction containers, researchers were able to determine the full sequence based on the fluorescence that they saw from each of these 4 reaction containers. So as you can see here, like A is a green color, T is a red color, and from that they were able to find the sequences. So then another very important concept is PCR, or the polymerase chain reaction, for DNA replication. And so it's a technique that can be used to generate many copies of an identical piece of complementary DNA, so also known as cDNA. And the most common type of PCR that is usually used is RT-PCR, which RT stands for reverse transcriptase. And it refers to that conversion between RNA back into the complementary DNA using that reverse transcriptase enzyme. And PCR uses primers. So the main things that we need for a PCR to the technique to be done is primers, Taq DNA polymerase, DNTPs, and double-stranded DNA. So this might just be a review, but the steps of the PCR is first denaturation. So this is usually done at really high heat. Um, I'm putting an example here for 95 degrees Celsius. And it's used to it's used to break the double-stranded DNA into single-stranded DNA. And also, specifically, we use Taq DNA polymerase because it's an enzyme that can withstand this high heat. So that's usually why specifically we use Taq. And then the second step after denaturation is annealing, which the temperature is then decreased to about 50 to 56 degrees Celsius. And that starts allowing the primers to bind to the single-stranded DNA, since they weren't able to before when it was in that double strand. And then the third step is elongation, where the temperature is then slightly increased to 72 degrees Celsius to allow DNA polymerase to extend the primers using the DNTPs that are given. So those are the different nucleotides, and it's at a better temperature, or the optimum temperature for then the polymerase. And then after this, there would be hybridization of those extra strands. So then they would form back into their double-stranded DNA once they kind of hybridize with each other. And then the cycle repeats according to how much DNA that an individual is trying to replicate. And the amount of DNA would be 2n, where n is the number of cycles that you are running that PCR. And so, as you can see here, this is pretty much at the bottom, it's a diagram of how PCR works, where you have that double strand, then it's broken into the single strand, and then you have the primers being able to attach to that single strand. And these are then elongated, and as you keep going through the cycles, they, keep doubling.